Welcome to visit our GeneScan Precision DNA Analysis System. The GeneScan Precision DNA Analysis System is a quick gene identification system, integrating and streamlining the classic PCR technology and DNA fluorescence staining technology. The whole process can be accomplished in less than 1 hour. There are 4 modules in this DNA identification process. 1. DNA purification, 2 to 15 minutes. 2. PCR amplification, 25 to 35 minutes. 3. Fluorescence imaging, and 4. Results interpretation, 1 minute. In the current first generation of the system, dependent on the source of the specimen, DNA purification procedure varies. For saliva samples, genomic DNA can be released right after the lysis treatment with the GeneScan sample buffer, and which can be used immediately for downstream assessment. For blood or fecal samples, a brief magnetic bead-based DNA purification is necessary to remove the potential inhibitors for downstream analyses. The incoming second generation of GeneScan Precision DNA Analysis System will automatically extract and purify the DNA from the specimen by embed magnetic DNA purification module. The DNA sample mixes with GeneScan enzyme mixed by simple pipetting, and the mix is loaded on each well of the GeneScan chip according to the layout map provided by the kit user manual. Turn on the PCR unit, open the top lid, and put the chip on the sample holder. Then close the top lid. Go to the main page of the PCR unit LED screen. Select the designated program and amplify the target gene. The process can be done in 25 minutes to 35 minutes. Once the PCR is finished, click the Stop button on the bottom of the LED screen. Open the lid and take out the chip. Turn on the fluorescence imaging unit. The main menu page will automatically pop up. Click left arrow to open the chip loader. In the same time, the left arrow changes to left arrow. Put the chip on the loader. Click left arrow to close the loader to side. Click Human Portrait symbol on the left, and Specimen Information window pops up. Fill in the necessary information with the virtual keyboard. If the specimen information has already been stored in the database, once the specimen ID is typed in, all the remaining information is filled in automatically by the software. Click Finish button to save the information, and close the window. Click the center of the circle on the right side of the screen. Initiate the imaging process. The circle turns green once the fluorescence image is taken, and the result is interpreted and reported as PDF format. Click the settings on the left to exit the imaging software if no further chip is expected. The report is generated automatically and stored in the default location preset by the administrator. If network path is defined and set, the report can be accessed via network shared drive or cloud data center. Email or phone notice automatically sending to doctor or patient if set up. The report format can be customized based on the customer's request. GeneScan Precision DNA Analysis System is designed for quick and reliable gene identification for any DNA specimen, such as human saliva, animal fecal sample, or agriculture specimen, etc. It can simultaneously detect up to 50 samples in one GeneScan chip less than 1 hour. Short window time, accurate results, low cost. Gene identification is affordable for everyone, the poor and the rich.